Good evening, friends. When it comes to the clinical trials, we are almost always confused as more our time is actually spent in the clinical practice, especially those our colleagues and clinical practitioners who are not full-time researcher. They are not very clear about the how the trial is being designed and what purposes it is. So we start with the our one and most important foundation to know the what is research. So most pragmatic definition of research is to known unknown things, and then finally we crack the known. The unknown things, what does it mean? In universe there is always something is there. It is always some information or some phenomenon, whether it is a true or not true. If you even don't know what you don't know, and suddenly things you reveal a truthfulness of the universe, that was a by chance events, and that is typically invention of the chest, invention of the X-rays. That is by chance. Nobody actually planned the any trial over there. So that unknown, if you crack that fact, that would be a by chance. So typically here in the second circle, where the known unknown, that means there is a research question and you want to answer that question. You know what you don't know. And ultimately, you try to reveal the truth which is already existing in the universe. The question, your research question, is in your favor or not. That is a most pragmatic way to understand the research. Classically, it is defined as quest for knowledge through systematic and scientific way on a specific topic aimed at discovery and interpretation of new knowledge. The same concept which I described in a more pragmatic way, and this is a classical scientific way to define research. Now, from here to move on to the research, what error in general can happen when we plan to answer our research question? Obviously we conduct a research. So what could be a category of sources of errors in the research in general? Typically a sampling error, it can happen, and sampling error can happen at any step when you are framing your research. That is, you're framing the a wrong selection of the patient or wrong selection of the subject based upon the framing of the research. That can, you can say it is a frame error. And second is by chance or chance error. You don't want to commit the error, but somehow while recruiting or selecting your subjects, by chance actually you selected something which interfere in correctness of your result. That is a sampling error. And non-sampling error, during your study, subsequently, of course, you took a correct sample which is commensurate to your research design, but it can be a non-sampling error, like a response error, measurement errors, performance error. These are the two broad classification of errors. Eventually, because we started our discussion from the research, so we need to know if your research is not cracking the correct or accuracy of your answer to your research question, then these are the possible source over there. These are the two specific categories. Now, during the flow of your research journey, we can identify the certain steps where your error can happen, or biases. What are biases? Because of some error, the biases occur. So that at what steps the biases can happen. Say, for example, you target population baseline state and you allocation of your subjects or your patients or this thing into the intervention group and control group. I'm talking about the very, very specific study design. Of course, subsequently we discuss what are the different studies or designs. But here the classical is a randomized control trial or some control trial. Let's see the random how you can overcome. There is one is a control and one is the intervention group. So when your allocation of your subjects are happening at that moment, you can actually have a selection bias during that process. How do you define that? Systematic differences in the comparison group attributable to incomplete randomization. That means these two groups are not identical in representation except the intervention has been done. That is a primarily initial sampling error related bias. So at this moment, you right from the your beginning of the research, you don't want to allow a process or error can lead to the selection bias so that this bias can be overcome by the randomization. So that is the first step where bias can happen. Of course, you overcome that bias. You randomize and nicely randomize in our subsequent part of discussion. We will have a great discussion about the randomization and what are the different ways to randomization. Anyway, you randomize it nicely, identically. That means only the your intervention is different. Rest of the things is similar in your population, whether it is a control group or the intervention group. Then you expose them to the intervention. So, of course, you are exposed, that is the intervention group, and of course, the rest is the control group. Depending upon the your research question, if it is a drug trial, then the placebo and all these things. Let's see how the so if other group, if you're not doing anything, then there could be a possibility of performance bias. That means you may treat intervention group differently from the control group. So systematic difference in the care provided apart from the intervention group. So that can be overcome by blinding. So that you can blind, and one of the method for blinding is the placebo. So if you are giving the drug to another subset of the patient should get externally appearance similar kind of this thing, but not actually the drug over there, so that your performance will not be confounded. Later on, you follow them up. Of course, you collect your data. At that level, biases can happen, and you can call is the exclusion bias. During follow-up, you may not collect some data. You feel whatever, knowingly or unknowingly, you remove certain data from your trial or you just exclude that patient. So typical, usual this thing is if you're not able to get or if you're not able to follow up, you remove from there. So that is again lead to the exclusion bias. How do you define that? Systemic differences in withdrawal from the trial. So they may or may not actually affect the result of your trial, but of course, this is again a biases which has potential to affect the trial. There may be a certain trial design for withdrawal also. Again, we'll discuss in the a subsequent part of discussion, but that would not be your systematic bias. It would be a randomization withdrawal. So that is a plan of your study. That is, in fact, you design that study. Otherwise, if you don't design and you exclude, then it will call the exclusion bias. Of course, now you collect the data, you analyze it, and certain outcome happens. At that moment, detection bias may be there. You may not be able to detect it properly during your analysis, and that is called detection bias. So these are the steps where the biases can happen, and your research question can lead to the. If not necessarily, this bias is always lead to the wrong conclusion, but that has the potential to conclude wrongly during your research trial. That's why when we are going for the systematic and the good quality of research, we have to take care of all the biases so that whatever we get the result, we believe in that result and we conclude it in an accurate and correct way. That means closer to the truth. We talked about the certain biases and there is a remedy for that. One that is a universal bias for any clinical trial. And unfortunately, there is no remedy for that. Very interesting. What is that? A universal bias, this is a phenomenon we'll discuss, is it is a study subjects get better clinical care than in routine clinical practice. So if you put somebody in the clinical trial, they will definitely get a better care than what you are doing the routinely kind of this thing. Say, for example, a certain population, you are trying for the certain drugs and you are keeping the record of your adverse effect of the drug. Or say, for example, analgesic you are comparing. So your data collection is having a part of this thing, how much change in their hemodynamics, what are the adverse reaction happening or undesirable this thing, because you have to note it down. So you pay more attention to meticulously all these patients. Probably in your clinical patients, same patient when you're treating in the clinical practice, will not pay attention to these parameters because that is not part of your research. As well as the research is having a some kind of your performance. Eventually, you have to present it, you have to accumulate it and going to reveal it somewhere in the journal or in your meeting, in your presentation. So what we call is your performance bias in terms of the Hawthorne effect, where the observational changes behavior. This is more related to the human behavior, human psychology. If somebody is seeing you, then your behavior would be a change more nicely kind of this thing is. We all know the people having a good profile picture in the WhatsApp profile, and probably their Aadhaar card is not having that much very impressive photo over there. What is that? It is a purely Hawthorne effect. When subject of an experimental study change or improve their behavior because it is being evaluated or studied. And clinical trial, we ultimately, whosoever is the researcher, whosoever is the collecting data, all knows. Even you can blind the certain observer. They don't know what are the patient in which group. That blinding can be done, but definitely they know that it is a research. The subject know because you took the consent from the subject. Subject also know he's a part of the research. Of course, he's a blind for the which group he's assigned to, but he knows he's a part of the research as well as your data collections. So this is this, and especially prospective trial. A retrospective trial, nobody knows. So from this aspect, retrospective trial is having a immunity against this biasness, a Hawthorne effect, because that clinical practice is going on irrespective. Nobody knows who will going to see their data and their performance. Later on, you collect it. From this aspect, your observational trial is better. From this observation, of course, that your retrospective trial is having a number of the problems so that you cannot conclude correctly. But this aspect is overcome over there. Okay. Come back to our classical types of error from the research outcome. The first is the type 1 that we call is the alpha error. So rejecting the null hypothesis when it is true. What does it mean? Null hypothesis means there is no effect over there. If you are doing some drug trial, so you start with that this drug is not going to affect the outcome. This is a null hypothesis. If you reject this null hypothesis, that means establishment of the alternative hypothesis, then you reject the null hypothesis. If it is a correctly, then it's okay. If you reject null hypothesis when it is true, That means null hypothesis is the A drug is not actually giving benefit to the patient. But your research showing in reality it is not there. As I told you, there is some truth in the universe, but your research showing this drug is helping to the patient in that particular disease. It is a wrong, it is an error. This is called the type 1 error, rejecting the null hypothesis when it is true. So, what happens? That means if your research is showing this drug is effective in that particular disease, you keep on giving the drugs to the rest of the patient. So, it would be unnecessarily intervention. So, it will give you a wrong, a more burden on the unnecessary intervention. May or may not cause the harm. Of course, drug property can cause the harm, but your action is not actually harmful from the research point of view. What if other way around? Other way around means your drug is actually effective in this disease, but whatever way you conduct the research, we have discussed that so many biases and errors can happen at various steps of the research. Somehow you concluded this drug is not effective in this. So what you actually did, accepting the null hypothesis when it is false. That means in reality, null hypothesis false means drug is effective. But your research concluded drug is not effective. And what will you do? Your research will end up with the which situation? Your action would be you not give to drug to the patient. Technically, you are depriving that patient from the drug which is actually effective, but falsely or erroneously your research showing error in your research. But who will be ultimately victim? The patient is a victim. So this is called fatal error. So first one is the missed alarm, and second is the fatal. So first one is the overdoing, may or may not cause the potentially harm, but second is that really your research is harming to the patient because you concluded this drug should not be given. So these are the two important, I guess, are all the viewers actually understand, and this is very very important, an important foundation for our research, most commonly asked question in Viva and all the researches. So I guess of course the subsequently I'll give the example also. Type 3, the type 1 is alpha error, type B is beta, type 2 is beta error, type 3 is the gamma error, correctly rejecting null hypothesis for wrong reason. So your everything is correct. Your conclusion and all this thing, your explanation is wrong. You're given the wrong reason. That's okay. Ultimately, your paper would be a little bit, a quality will be lower down. Your based upon your scientific data and facts, but obviously it is not that much fatal to the patient. Similarly, type 4 error, the incorrect interpretation of correctly rejected hypothesis. Again, your interpretation is wrong. The previous reason you gave the wrong, but your interpretation wrong. But eventually, the action is in the favor of the what exactly reality. So these are the 4 errors which we should know. So if we make a 2 by 2 table, like given the null hypothesis is really true, and from your decision based upon a random sample or research actually is rejected. Null hypothesis is true, and you rejected null hypothesis. That is the type 1 error, alpha error. If null hypothesis is true and you do not reject it, actually you accepted the null hypothesis, which is correct. So your decision is correct. Similarly, this upper part is in reality. So in reality, null hypothesis is false. False means intervention is effective in reality. But your research is also showing it is then correct decision because you reject the null hypothesis, which is false. Null hypothesis is false and you do not reject it. That means you accept it. Actually, it is false. And this is a type 2. This is a fatal error. So I give you example over there how important the sample also when we give the examples. Just consider one scenario. Out of 10 smoker, 9 have carcinoma lung. This is the one situation. So very likely you will have a strong conclusion with this one statement. Very likely smoking causes the cancer lung. This is actually hypothetically just to understand when 9 persons are having a cancer out of 10 smokers. Second scenario, suppose 10 smoker having uh, out of 10, 5 will have a carcinoma lung. Will you say with same confidence the smoking causes lung cancer? Probably you may not be. So in the first scenario, 90% of the person is having association with the smoking, then you are very much confident to say it. 50% you may not be. Okay. When out of 10, 1 has cancer lung, that means 90% is not having. Will you able to say confidently same amount of confidence is for the negative aspect? That means smoking does not cause the cancer lung. You will not. What I mean to say, when occurrence is actually happening, even the small 10 number and 90% is having a cancer, you will be able to say it confidently smoking causes. When 90% is not having out of 10 only, in the first case also 90% having, in the last case 90% is not having. Same number, but just for the 10, 90% or not having, you may not be able to say confidently. What if this 10 number convert into the 1 million, 10 lakh? So 90% of, or say for example, 10,000, 90% of 10,000 is not having cancer, then probably you will say, oh, yes. Again, the percentage will be 90%. If 1 million, 10 lakh, if you study 10 lakh and 90% is not having the cancer, so 10 lakh of the 10 lakh smokers and 90%, then you are much more confident to say it is not causing the cancer. Are you getting my point? When there is null hypothesis going, when your trial is showing the no advantage, to conclude that you will have a much larger number than the first one. Of course, first one is also having a some kind of a number. Same thing, if 100 smokers are there and 90 is having a, you will be much more confident to say it. Okay. So similar thing, this is the why your power is important when you're giving in reality your null hypothesis is true, but your research is showing the alternative hypothesis, that is a false alarm, overactivity, and misalarm. I already discussed. Now you able to, power is nothing but your number of subjects or number of recruitment or number of your observations depending upon the name. So it is a probability that the test reject a false null hypothesis. That means it will not make a type 2 error. So power having a very, very crucial role when your research is showing this drug is not effective. And that's why you require a larger number. That's why the beta error and the number are related to each other. And that is the equation is power is equal to 1 minus beta. So if you want to have a lower beta, then you have to increase the number of your observation. Now from here, these were the certain important and foundations for clinical research. Now I am going to the design classically, how the things are happening, how the research is actually design. So clinical research, immediately we divide into the two categories. No intervention, intervention. We label it as a no intervention means observational study. Observational study can have whether the comparison or comparative group is there or not. If it is yes, then it is your case control or cohort study. That means, of course, there is a two group to compare it, but it is a observational. Researcher is not going to affect like a flowing river. You are just seeing the river from your bank of the river. You are not doing anything for there. This is, and if you can compare, then it would be a comparative study, case control or cohort. We'll discuss difference between. If you cannot or you don't have any comparative group, you just study the one thing only. Like if you are seeing the two rivers and comparing, then it would be your a cohort or case control. Let's see, depending upon the we'll discuss subsequently. But if you are seeing only the one river, only and try to study all the properties and all these things, then it would be a descriptive study. Previous one is the analytic, the descriptive. Say for example, descriptive is an epidemiological study. You research the how your distribution of the disease in your population. You study the what are the clinical manifestation of the one disease. All are descriptive. There is no comparison, and you're just seeing. You are not actually modifying the disease. That's why these are the descriptive study. Now intervention. So intervention, also called as experimental. At this juncture, I would like to clarify to our all the viewers, it is an intervention in the research point of view. It may or may not be actual surgical or procedural intervention, because people are really sometimes very, very confused. When we do some surgery or research, that is intervention. That is intervention in your routine language of the clinical practice English language. In research, intervention means you are altering the flow of your studies. You are doing something over there. It may or may not be a certain procedure. It may be a drug. It may be even the surgery is happening or some procedure is happening. It can be also observational study. You are just seeing the how after the surgery the outcome happens. So intervention is being done, but research is no intervention. Similarly, without surgery, if you are just dividing two persons, two groups over there and observing them, you are dividing the previous. You can ask then there would be a comparative group in the case control or cohort study, but that is the already naturally made cohort. Here, you yourself selecting certain people and dividing into the group depending upon their behavior and all these things. When your intervention happens, then it would be a intervention or experimental study. If this if this you your intervention in a systematic randomized way, that means you are having we'll discuss what is randomization rather than your whims and fancy or your just arbitrarily your points or your anything based upon the say for example just odd or even number or days of your week and all these things. That is not a proper randomization. That is also you are dividing into the group. That is the non-randomization. But obviously you are dividing. That would be intervention. So if randomization allocation happens, if it is yes, then it is a randomized control trial. Very important, and that is the major things which we'll discuss. And if you are not randomizing it, you are just giving an example. The people come and you want to study the two surgical procedure for the same problem. One surgeon prefer A surgery, other surgeon prefer B surgery. So you just take the patient and divide among these two surgeons. Of course, you are dividing it. And why you can't randomize? Because the whosoever is willing to this thing, and if you are not randomized, because they are coming and you are sending. If they want this type of surgery, they go to the B surgeon. If they want that type of surgery, their preference, they go for the B surgeon. You are not systematically. That is a non-randomization. But still intervention is there. You are dividing over there rather than automatically it is going. So it is a no rand, non-randomized trial that is also comparative. And you are intervening. That's why it is an experimental intervention. So this is a very, very simplistic and the basic foundation. At this stage, you should be very clear. Otherwise, you can go back and see the video. At this moment, you should be very, very clear because subsequent discussion is based upon that. Now the question comes, who can decide what kind of, whether we should go for the randomization? Always randomization is better. Sometimes randomization is not possible. All these things, sometimes it is unethical, not feasible. So what are the points? So, and how you can design? So factor to consider in selecting a clinical trial design: study population indication, treatment duration, carryover effects, costs and logistics, patient convenience, and statistical consideration. So there are so many things. It's very easy. Everything cannot be planned for the randomized controlled trial. So there are so many things, and that's the reason today we are discussing the different clinical trials. Now coming back to the one arm observational study. As I told you, observational study, it can be a group, a comparative or analytic. So one is the cohort and case control. They both are analytic. Cohort is the prospective future. That means you selected a certain exposure. So you selected, expose them to the certain kind of a thing you are studying, and you go for the outcome. You are not dividing them. Let it happen, and over the time period, you divide into the two groups for analysis, not for the exposure. Case control, other way around, outcome already happens. So you are having those persons where outcome already happens, and now you go back and try to see what happens. Just I'm giving you an example. In a certain society or maybe a small town, and this thing is, you are having some hypothesis like those people who are consume more social media having a higher rate of depression. Suppose if you are studying that cohort study, then you prospectively see the person, you enroll them and see the who are going to consume more and more social media, who are not consuming the social media. You are not governing who are going to consume. They just do in their own way. And you follow them up for a year, 2 year, 3 year, 5 years, 6 year, 10 years. And after the 10 years, you find those who are not having depression, those who are having a depression, there's one way. Or those who consume the social media up to a certain point, every day kind of you can make the matrix, and those who are having a less, and you study over there. This is a cohort study. So the exposure and then outcome happen. Other way around, as if now you cross sectionally in the society and selected all the depressed patient or not the depressed person, and go back how long they are consuming the social media, how much hours is there, how long it is there, which is already happened. And this is called case control. So outcome happen and you are going back for the exposure. That is a retrospectively. Another comparative cross-sectional, as if now, right now, how much is the things happening cross-sectionally, just transversely at this moment, how much percentage of the person consume the average 6 hours of the social media, less than 6 hours of the social media, transversely, just maybe a survey. That is the cross-sectional. All are observational study. So exposure and outcome happening simultaneously. How many is less than and how much depression? There's completely transverse section over there. So one thing you observe, case control and cross-sectional study, one common phenomenon happens, your outcome and exposure already happens. So you're not able to see whether they are having actually depression, that's why they are seeing the more social media, or after seeing the social media, depression has developed. That you can't, and that's the egg or chicken theory over there. It gives you a just association. It may not be able to give you a cause effect relationship. Whereas cohort study is this property to establish a cause effect. And of course, in today's we study, our session, we'll not discuss about that. What is the cause and effect? Of course, if you're going for the further research, then you come to know how we differentiate between association versus cause. Because I'm just giving a simple example. A people who is already having a tendency for the depression, that's why they are consuming more rather than the social media leads into the depression. That one hypothesis. Another hypothesis, because they are seeing the more social media and then depression happens. That cannot be answered over there. And that is the most important here is the cohort study. That's why the most lifestyle related this thing, diseases like diabetes or hypertension and so many cardiac diseases. So different society actually keep on conducting a trial for the 5 year, 10 year, 15 year, 20 years, 25 years, 30 years. So it's a long term kind of this thing. So that's why you are sometimes getting a contradictory results. Initially, cholesterol is bad, then it is not that much bad. Then this thing, why? Because as the time passes, the precision happening in the research. Okay. Interventional, non-randomized, randomized, we already trial. There are different way to classify design also. That is which I told you is a completely a more and more relevant and very basic classification. Sometimes people can also divide the trials based upon the their basis of method, on the basis of awareness of the participant, on the basis of magnitude of the activity. That is also you can divide on the basis of method. Example, randomize, that is the one aspect. Awareness, like unblinded, blinded, or active drug control or placebo drug control, we discussed. You can divide in that way also. Magnitude of activity, superiority trial, equivalent trial, non-inferiority trial, dose response relationship. So that is also one of the way for the classification of the trial. And this is not the either or kind of this thing is like intervention group, randomized control trial. Randomized control trial can be planned for the superiority. Randomized control trial can be planned for the non-inferiority. Randomized control trial can be planned for the equivalence. In this way, we classify the we classify the different trial designs. So, in nutshell, what are the salient feature observational versus interventional studies? Like a study design, observational participants are not assigned to any intervention. Obviously, an intervention group, they are assigned to one or more interventions. Goal to observe and collect data on participants. Here to evaluate the safety and efficacy of intervention. Types of study, cohort study, case study. We discussed a lot about that. Here, therapeutic, preventive studies in the intervention group. Types of intervention, obviously done in the observational study. What can be a types of intervention? That's I wanted you to again re-attention of your this thing is it can be drug, it can be device, it can be surgical procedure, it can be behavioral interventions. So intervention doesn't mean some motor procedure is going on. Observational study, less expensive cost is a general statement. If you are planning for the observational cohort study, then it may be very costly because it goes for the years together. Otherwise, in general, like a case control or point prevalence, it's very easy. Intervention trial, more expensive. Time, less time consuming. Again, little bit exception over there. Less time consuming. Which study observational? Cohort prevalence, point prevalence, transverse kind of study. But longitudinally, cohort studies are very, very long and more time consuming in general intervention studies. So I gave you a little bit glimpse of the cohort study. So again, we are back and forth. We're discussing a trial, then the comparison, then again go back to the observational. So now we complete for observational and go to the interventional. So observational cohort is you already selected a non-smoker and smoker. You are not offering to the people you will smoke or you will not be. You just select, let them do their activity in their own way. That's why it is a observation. And you follow them up for the years, and finally outcome happen. Then you try to compare the outcome. Those who didn't smoke, how many have your outcome? Say for example, cancer, and those who are already smoke, and how many? And then you compare over there. So if we what are the salient feature of the large population of patients who have a specific condition or receive particular treatment over time, compare with another group who has not been affected, observational study, not as reliable as the randomized control trial. Why? Because we paid attention to the those who are smoking, those who are not smoking. But rest of the things, there may be some are the low socioeconomic, high socioeconomic, uh, they are actually uh, they may be they may be residing in the different places environmentally. Few people are maybe exposed to the pollution over there, and though they may not, they are not known smoker, but still they are exposed to the pollution. You are not. So apart from your intervention, rest of the thing is still not similar between these two groups. That's why your result is not that robust, reliable as randomized control. Case control, I already told you. You start from here, not from there. Outcome already happened and go retrospectively, go back over there. And now compare from the previous happens from here, compare and draw the conclusion. So, lot of patients who are already having a specific condition, this is very easy in terms of their complexities, cost, everything is very easy over here. But lot of problems here. Lot of biases can happen, recall bias. They are often rely on the medical record and the patient recall, because you are taking the history or going back to the your record, in what circumstances, what records happening. And sometime I gave you one example over there. When we are comparing a data of the code orange, which is meant for the transfer of the patient from floor to the ICU, and we are keeping the data for that. And later on, we are analyzing what happened to them. So I collected all the data and divide into two, of course, not equal group, but just for to convey the message over there. 10-20%, maybe a say for example, 20% patient actually didn't transfer to the ICU. Based upon the certain parameters, they remain in the flow. And rest of the people transfer into the ICU. And we follow them of what happened to them. Those who actually transfer to the ICU, say for example, 80 out of the 100, they 12 or 15 patients actually died eventually. Of course, they were having a disease, that's why they died. Somehow, those who remain on the floor, no one has died. What conclusion we can make? If the patient getting sick, don't transfer into the ICU, because there is some mortality rate over there. Those patients is having a zero mortality. That is the problem with this case control. Outcome already happens, and then you retrospectively try it, and you may lead to the wrong conclusion. Statistic relation does not mean that one factor necessarily causes other. I already discussed. You nobody knows egg first or chicken first. Okay, now we are going to the interventional trials. So uncontrolled trial, the single treatment, baseline, and endpoint. That is uncontrolled. You just compare over there. Another variant of placebo or controlled trial design. Here you are making the two groups. Okay. One test drug, another placebo. Second, maybe placebo and active control. Active control means already your um, standard of care is going on, and you can compare. We will discuss subsequently over there. So we already entered into the intervention trial, observational trial already. We finished it. Now intervention trial, I am taking a certain pockets and later on I'll make the dots to make you understand. So first you understood uh, uncon- uncontrolled trial. There's no control. You just one group and you study it. Second group you can have a control. Control maybe placebo, maybe active drug. We'll discuss, of course. So you can either you can have a placebo or maybe active drug to the another new drug. You can compare over there. When this allocation is randomization, that becomes a randomized control trial. If you are not randomization, then it is a pure uncontrolled control trial, but non-randomized. Okay. So here, once we are using the placebo or active drug, it would be a control. Doesn't mean randomization only. It can be randomized. It cannot be randomized, depending upon the whether you are doing the randomization or not. When you are introducing the placebo, there may be one step also. Why I'm selecting all these things? When you read the trial, you may find a small this variation in your research paper, which is published in the journal. So it will help you what exactly they are meaning about the certain nomenclature. Placebo run-in design. That means you screen the patient and all these things. You give the some placebo for run-in just to test your placebo is actually working for you or not. So if it is unsatisfactory, then you drop either in that patient or the placebo or those particular patient. And if you find it satisfactorily, now you whatever this thing now you randomize into the two groups and give the active give the research drug or new drug and the B for the placebo. So what I here emphasize on the placebo in run design. Nothing very complex here. Just one step before randomization. You give them placebo and see if it is working satisfactorily. If it is not working certain satisfactory, then you remove for the still you are not doing the randomization. That's why the intention to treat is not applicable here. Intention to treat once you randomize it, and then you continue the rest of the. So comparison trial, there are a number of different trial for the comparison as possible on the basis of magnitude of the active drug, superiority to demonstrate the investigational medicine is better than the control, equivalence, demonstrate the endpoint measure is similar, no worse, no better than the control, non-inferiority to demonstrate that the investigational medicine is not worse than the control, and dose response relationship is to determine various dose parameter including starting dose and maximum. So all are the when you recruit all these things randomized, then all are randomized control. Okay. Quite often we see the superior trial and non-inferior trial. So I just submit here in a table superiority trial versus non-superiority trial. Primary objective here is obviously demonstrate the new treatment is better than the control or placebo. Non-inferiority is not unacceptably worse. So here keyword is not unacceptably worse. Means there is a certain provision for the acceptance here. That is the key. Once you understand this statement, subsequently you will be able to understand. Then you will be able to differentiate a superiority trial versus non-inferiority trial very easily. A rationale use when there is need of clinically meaningful improvement or outcome, like increased survival or faster recovery. It is used when the new treatment may offer non-efficacy benefit, fewer side effect, low cost, easier use, and just need to be a good enough. Because why we are going for the non-inferior trial? Because it is not going to really change your outcome. You are not going to the superior kind of this thing. Why you are doing? Because there may be a certain reason, certain drug is not a certain drug is costly, certain intervention is more complex, and if you are doing the less complex and the non-inferior, then you go for that. So more towards the logical thing and logistics rather than a real clinical outcome. Here control is placebo or active control. Always active control. Active control is a drug which otherwise you are giving. Otherwise, in your clinical practice, a part of treatment. If new drug and you are comparing this is the active control versus new drug. Placebo means completely pharmacological inactive. Understood? When you are doing the non-inferiority trial, that means you are proving the newer drug or intervention. Keep it drug here so that it is easy to understand. And giving the active treatment, then only you can, it is non-inferior to the active treatment. That's why non-inferior trial placebo cannot be done. Placebo cannot be used. This is very important. Null hypothesis, the treatment is not better than the control. That is null hypothesis, and you reject the null hypothesis. You start with the always negative. So you are starting, I am giving a B drug, A drug patient is already getting, and I try to prove the B drug is better than the A drug. Either you go for the B to placebo, A to placebo, and go for this thing is, but it is theoretical. Practically, you can't give the placebo when your treatment, or you can directly B drug to the A drug, which is the active control. And you make a hypothesis, my B drug is not much effective, same effectiveness, and research, you actually reject the null hypothesis. That means your newer drug is very much effective. And then you go for that. That is your basis for the hypothesis. Whereas in non-inferiority trial, the new treatment is worse than control by more than a predefined margin. This is a very, very key statement over there. In non-inferiority trial, we can't take mark to mark outcome. We accept the certain predefined margins just to make the things easy. If you are newer drug for the pneumonia or pneumonia you are studying, the A drugs, say for example, azithromycin you are giving, your aim is the B and the mortality, just assume it is a 15%. So you make the hypothesis, I will accept the 5% margin of it. 5% margin of it means 5% of the 15%. Even if it is a more, then I will take it as a equally effective. Less, obviously, you are not aiming for that because you are not aiming for the superiority. The margin comes for the worst outcome. So this 5% is called the margin. Okay. And what will be hypothesis is this drug causes more harm than the margin. That's, I'll make a hypothesis exactly opposite for that. And when you actually reject this hypothesis, that means your trial will be a really positive. That means this drug is non-inferior to the active drug. If you understand this thing is okay, otherwise keep one basis idea about that. Non-inferior trial always have a provision of margin delta. Otherwise, a superior trial does not have such kind of this thing. Okay. So pre-specified margin not required. It is required. I actually overemphasize this point. Rest of the thing, sample size often larger, maybe smaller, two-sided test typically, because when you are going for the superiority trial, because you started with the null hypothesis, because it is not equal or maybe inferior to that. So that's why this trial can land up to the inferior, can land up into the superior. That's why you are having a two-sided test. Whereas here your aim is to not inferior, one-sided. Okay. Now the question comes then, why we are planning for the non-inferiority? Why don't we plan for the superior trial all the time? Because treating a new oral anticoagulant example for comparative study, but easier administration than the injectable heparin. Why we are planning all the time scientifically or maybe ethically not able to go for the superiority, then you go for the non-inferiority. Okay. So we already entered into the interventions trial, superior non-inferiority, control, and all this thing. Now I am taking the most important aspect here is the randomization. Very, very important. Subsequent 3, 4 slides, very meaningful to understand, to read the paper, very meaningful in your exams. And if you are a researcher, then obviously it is the most important. Randomization initially divided into the fixed and adaptive. Fixed and adaptive means fixed means your rule of game will be defined right from the beginning and it won't change. Adaptive means you keep on changing your rules. We discussed. So fixed, again, fixed can be divided into the simple randomization. That means patient allocation is based on the single sequence of random assignments. Computer based numbers you can use. Just giving the example, tossing of the coin. Of course, it is not the standard way for the single randomization, but it is the example of the simple randomization. You toss head and tail, and you keep on dividing into the two groups. This is actually a true randomization. Each and every person you are taking into account into the probability of the two group. This is a simple randomization. Block randomization is patients are first allocated to block, then randomized with the block. What does it mean? The previous one, suppose 400 persons are there, your subjects, and you are try to allocate just an example is tight sugar control and not tight sugar control, liberal control, or class whatever traditional control, and you are simple randomization. Every subject coming to in front of you, you randomize computer based, but technically that same tossing the coin. Block randomization. Second scenario, all those 400 in front of you. What you are doing is you take out 4% or 4 subjects initially. First, it would be in the even number. Now this 4%, you randomize it. Okay, head tail based upon. So what can happen? The first person can go to the A group. Okay. Second person, if he goes to the A group, then you need not to randomize these two. These two automatically will go to the B group. This is one scenario. Second scenario, if first person goes to the A group, second person goes to the B group, third person goes to the B group, fourth person need not to randomize, automatically it will go to the A group. Third scenario, if first person go to the A group, second person go to the B group, first person A group, again the fourth person. So what happens in the block randomization? Last person randomization does not require. Did you observe what is happening over here? You can equally divide into in the previous one, when you are tossing the coin and 400, you may not be able to achieve 200. Very likely you will have a 180, 220. Whether it matters or not, that's a different issue. But here you can meticulously go for that. And block itself can be randomized. Means first time you took the 4%, second time you took the 6%, third time you took the 6%, then again 4, 6, 4, 6, 4, 4, 4, 4, 4, 6, 4, 6, 4, 6, 4. So block itself can be randomized. Now, when to use? Especially block randomization, your subjects or N or patients number or study population, if it is less, because less is giving you opportunity to the, and where one point, and second is you want exactly 1:1 distribution, then obviously go for the block. Okay. But if you compare between these two groups, which is a really randomized kind of this thing, actual randomized. First group actually randomize every person. Here what happens? There is probability. Suppose if you are taking the 4%, there's probability 2% will not be randomized automatically, because the whatever happened, fate is the initial 2 will decide their fate, and the last person will not have his fate. His fate is purely dependent upon the what are the fates of the initial three. He will not be subjected to the randomizer. He will be always allotted to it. Yeah. That's why it is not that much complete randomization like a simple randomization, stratified. Patients are grouped according to the prognostic variables, then randomized into the separate strata. By definition, this is, but I am giving you example so that you can understand. You make the cluster one and then randomize. Suppose you want to study uh, how uh, how the ICU practice for the pain control in the infusion form. Just giving example, you control the pain with the infusion kind of form. This is it, and you want to study across the country. Now, when you want to study the across the country, and you want to make the conclusion, but the point is there may be a corporate ICU is different from the AIMS ICU. AIMS ICU is different from the medical college ICU. Medical college ICU is different from the non-teaching hospital. Non-teaching hospital in the metro city is different from the non-teaching hospital in the town. Corporate in the town is different from the corporate in the metro hospital. There's so how you can there is possibility you can randomize all these things. Possibility is the A group is having most of the ICUs of the uh, maybe a non-teaching government hospitals. Most of this thing is then probably if you are studying that certain feature and it's actually affect over there, then probably you will get a wrong information. What you can do? You can collect the similar. I'll divide into the 4-5 group of the ICUs. Metro cities, corporate, 10-15 ICU. Now I randomize among them. Metro cities, medical college institute, 2020. I'll this thing is B grade town, non-teaching. All the B grade town across the India, non-teaching, maybe a thousands or whatever this thing is. I'll divide them into different group. In among those, this thing only. Everybody will be divided into the A and B group. So you will see A group is having a equal kind of a representation of the different group of your study population as a B. So whatever will happen, and these are the points which has potential to affect your research question and the result, and you make them equal over this. This is a stratify, and this process stratification, when you make the group stratify and then you randomize it. So again, but it is not as complete as the simple. Understood? Adaptive. But in all these three, the rules of game is already decided before the game is started. Right from beginning, how I going to make it. Adaptive is outcome adaptive or minimization adaptive. Outcome is patient allocation probability change throughout the trial according to incoming results, health outcome data, and all. We'll discuss a little bit. Probably you may be able to understand or not, but of course, I'll not pay much attention. I'll give you a glimpse of it, and you can research yourself. Here, what happens? You keep on changing. Your studying start. Your study started. Your study population keep on changing. The randomly you add new, randomly you remove it, randomly you add new, and this thing is because what is happening? What is outcome over there? And it is not static. Rules keep on changing. That's why it is adaptive, not the fixed. Minimization. Here, keyword is the outcome. Please remember, outcome adaptive is based upon your randomized based upon the outcome. Whereas minimization is you keep on recruiting the patient and you are continuing your research every time you look back of your study population, and if you find your study population is getting a little bit skew in this thing, then you change your rules and get the balance of your study population. Here, not the outcome. The equality of this thing is certain, because you compromise randomization based over the some factor which can affect your result. Now, don't ask randomization the supreme thing and all this. Depending upon your research question, sometimes you have to compromise the randomization because suppose you want to keep all the time exactly 1:1 male and female ratio. Then you keep on changing your rules over there. This time, if females come, then I'll go into this thing. That is a simplistic way. Otherwise, there if you take the two variables, then maybe a more. So this is a minimization. Allocation depends upon the previous allocation in a way that aims to minimize covariant imbalance. I hope understood. Here outcome. Here you just see this thing is using this method. The first patient is truly randomized allocation. For each subsequent patient, the treatment allocation is identified, which minimize the imbalance between group at that time. Okay. So right from the beginning, who comes? Giving example, you want to keep the 1:1. The first one who is coming in the A group is a male, and then you want to the next. Obviously, male goes to the B. So that's why. And what is the basis for that? Is the your already allotted population. So quickly, this is a diagrammatic representation. Block randomization, all the study you divide into the block one. As I told you, here is the constant block, but block itself can be randomized. How it can be randomized? 4, 4, 4, 4, 6, 6, 6, 4, 4, 6, 4, 6, 4, 6, and then further randomization. Stratification, I told you already you selected and you divide into here. Just example, these are the, say for example, metro ICU, and these are the town ICUs. And then you randomize among them, the group A and group B. This is your variable IC, variable adaptive kind of the based upon the outcome. So this earn is you keep on adding randomization and assignment according to the type of the selected at random. So outcome, how it's happening, and then you are changing. And here minimization, initial, randomize, you start the treatment. In between, you interim analysis over this thing, and you see your population. You find some problem is there, then adaptive randomization, interim adapt in terms of the whether you want to do it or not. Stratification, I already told you much about that. I just giving example why stratification is required. You want to 1:1 or uniformity, all these things. I'll not read all this thing here. Again, a message. First, stratification ensure the variables not confounded with the intervention, which is especially important, which I told you. Second, which is not the primary aim for that, but giving you opportunity, because now you divided into the giving example ICU of the B town, non-teaching, teaching, and you randomize among them. Later on, when you analyze, then you already having a subgroup analysis beforehand. If you want to express your data in subgroup analysis, then during the process of randomization and stratification, you are knowingly or unknowingly make the some subgroup, which is very meaningful also. So this is the second. If adequately planned, stratification allows subgroup analysis. Few more example of the design, parallel arm group. So you just design in parallel. Later on, crossover. So initial placebo and active group, same population becomes active, and placebo becomes active, and active becomes placebo. And obviously, this type of you require a certain cooling period and washout period and all this thing. Just I'm giving example. This also can happen. Initially, control become active, and then over the time period, you can go sequentially. Just an example, ICU related one trial. Long-term effect of flexible visitation to the intensive care unit on family member mental health, 12-month result from the randomized clinical trial. I'm not going to the clinical aspect, purely a research kind of a this thing is. So this is a long-term outcome and also cluster crossover randomized trial. The word cluster crossover. So in cluster crossover means initially those ICU were having a restricted family visit up to a certain observation period. Over the time period, those again they become a liberal, and those who are liberal become a restricted. So crossover happens. Here, see how the randomized. The ICU were randomly allocated to one or two sequence of intervention, flexible visitation followed by using restrictive visitation or usual restrictive and following 1:1 ratio. And how they randomize? Randomization was performed using block of random size. Block of random size. As I told you, 4, 4, 4, 5, 4, 6, oh, not 5, 4, 6, 4, 6, all are the even number. In addition, ICU were stratified. So one is the randomization. Again, the block itself is a randomized. And ICU were also stratified based upon the number of available bed. Categorized as less than 10 bed, more than 10 bed. Why? Because in the research visitation policy, if you are having a less than 10 bed, is having a different performance all the time. You can keep the patient's family all the time. It's having a different impact because 10 bed and the more busy ICU having a different impact, which may affect your performance. That's why they are stratified over there. One size doesn't fit all kind of a this thing is. 2x2 factorial design. Here what happens? You are studying the two new drugs and two placebo. So crisscross will happen. Very superficial. Of course, there's the four kind of a possibility over there. So placebo X, placebo Y, placebo X, active Y, active X, placebo Y. So ultimately, what are the, you are having the four things: X drug active, X drug placebo, Y drug active, Y drug placebo. In four thing, now you can make the combination and then go for this is called 2x2 factorial design. Matched pair. Again, there are interference in here in the randomization. What is the before randomization? You make the people matched first. Now you can ask you, then what is the difference between stratification and all these things? Stratification, when you initially one point over there, that means right from beginning you are recruiting the one point of time your subject. Like a previous, I gave the example, you're recruiting all the center in the kind of one. When ongoing types of recruitment and all the things happening, you match first and then randomized. Suppose if you are similar kind of this thing is you are taking the survey of the patient relative. So you match them who are the literate one or who are the professional one, who are this one, who are from the metro exposure and all these things. So you match and then randomize first. Withdrawal trial. There are the certain trial in a randomized withdrawal. Here, try to understand the randomization for the withdrawal, not for the recruitment. In a randomized withdrawal trial, participants receive a test treatment to which they respond for a specific time. At the end of a specific time, the participants are randomly assigned to continue the treatment with the rest test treatment or with the placebo. So it is other way around. You give the treatment first up to the specific time to the both the all the person. So you give the typical the cancer patient and all this in these type of trial. For the 6 months, everybody will get. Now randomly you remove the person. So you remove the certain person continue over there. What is going to interpret that is as if now we are not going into the detail of the what are the utility, how to go about that, what is the what is the advantage, when to go for such type of trial design. You just have an idea these type of the design can happen. So this is so withdrawal trial appears to be new to us that because most of the intensive care trials are not the withdrawal trial. To assess the response to the dose being stopped or reduced, to determine if treatment is still required, to discontinue participant in a study that have not responded to the medicine being studied. So you can actually, with the time frame, you can decide your outcome. 6 month treatment is good enough, or 1 year treatment is good enough, or all these things. You can go for that. Lastly, surrogate point, I touch a little bit over there. What are surrogate points? Surrogate points are variable, relatively easy, predicts rare or distant outcome, not itself a direct measure of harm or benefit, reduce sample size, duration cost, use when primary outcome invasive or unethical. Example, concentration to the about to the half-life curve, MIC, time above the MIC, microalbuminuria, tumor size, PF ratio. These are the surrogate. What does it mean? Here you want to see the effectiveness of the antibiotic, but you just see the how the MIC is getting over. Then you conclude A drug is better than the B drug. That is actually false. Some other day we'll discuss how to go for. You can say A drug achieved the MIC better than the B drug. There's a difference between them. Effectiveness, ultimately, your clinical outcome has to be there. It should be reliable, reproducible, clinically available, easily quantifiable, affordable, exhibit dose response curve, true predictor. There are a lot of things which we are using the surrogate, especially our viewers. I would give the emphasis again. Then whatever you read your paper, try to understand what they are actually looking at. Seeing the A drug causing less microalbuminemia or microalbuminuria compared to the B drug is having a different conclusion than saying A drug is effective in diabetic nephropathy. And most of the microbiological studies are based upon the surrogate endpoints. The surrogate endpoint may or may not reflect, and that is the drawback over there. Just few slides. Intention to treat analysis means all data on patients originally allotted to the different arm of the study, typically meant for the comparative trial. Of course, the different arm over there, and randomized control trial is the classical one where intention to treat. Intention to treat means your trial is like you are conducting a trial that is method for you. So you try to treat everybody who is coming into the trial. Treat means not the medical treatment. You treat them. You take into consideration rather than excluding them. Even after the randomization, later on, in the middle of trial, somebody withdraw the consent, which is all the time giving the privilege, but you will not exclude from the study. You still keep into the study and analyze it. Now the question comes, how can we go for that? Our endpoint is the mortality. He already withdrew after the 5 days treatment, where our trial plan is for the 1 month and all these things. I'll not go into the detail. There is a provision how to handle that data. Here is you keep the data, take into analysis, how to go about that, how to correct our there provision over there. Today we'll not discuss it, but that is the intent to treat analysis. Modified a little bit over there, we'll not discuss, but that is a we are reaching almost completion of our session today. Just you can pause it, take the screenshot. A typical study design for therapy randomized control is the best one, followed by cohort, case control, and case series. Again, please remember, I'll re-emphasize in our last slide over there. Your research question and your clinical parameter, whatever, that has to be meaningful. A nonsense thing subjected to the randomized control trial and giving a conclusion to over there may not be really excellent in the clinical practice. Your tool is excellent. Randomized control trial tool is excellent, but doesn't mean if it is the good tool used in the bad way, give you a bad result. So just all the time quoting this randomized control trial result into this thing, randomized control this result into this thing, and randomized control trial is the apex of the quality of the research. Statement is right, but interpretation is wrong. Your research question, research method is equally important than the randomized control trial. Diagnosis, prospective line, etiology, prognosis cohort is a more important prevention randomized and cost economic analysis. So this is a last slide where you have to keep the common sense along with the research. There are so many ways to know the truth. As we say, there are so many ways to nirvana. So there is so many ways to get the truth. Research is considered to be a most reliable in the scientific way, but doesn't mean all the time tool is excellent, but your method is wrong. Sometimes you don't require a tool that I'm giving you example, a single case report of thalidomide causing the phocomelia. The drug is withdrawn over there. Drug was withdrawn. So it is not necessarily all the decision will require a randomized control trial. Of course, the common sense has to be there. So with this, we end our talk. And message is education is not the learning of the fact, but training of the mind to think. That is the famous quote of Einstein. Thank you all.